Hello, my name is Ruben McWilliams, and welcome to Data Science with Ruben. Today I have an exciting video for you. It's detecting brain tumors using neural networks. Cool, so I'm going to be presenting a paper I found, presenting the code from it. So full credit goes to the people who wrote it. So this is it, and I'm going to be pasting the link in the description. But I basically am just going to be explaining the code from that paper so that beginners can understand it. So I'll be explaining in more detail, making things clear. Okay, great. So first I want to talk about the data set. The data set contains MRI scans converted to NumPy arrays. There are two classes in the data, yes and no. The yes class are MRI scans of brains with tumors. The no class is MRI scans of brains without tumors. So first, you need to import the required libraries. Okay, so just a big recommendation here. If you're new to this stuff, try to use Google Colab. I'll put the link to it in the description, but basically go to the link, then go to File, New Notebook. It'll create a new notebook for you. You can create cells and start putting code in here. And the main advantage of Google Colab is that it comes with like all of these libraries already installed, so you don't have to spend all the time like using like pip, which is a package manager for Python, for those who don't know, to like install these libraries. And TensorFlow is the most difficult one to install. It depends, the instructions are different depending on what operating system you're running. But actually, Google Colab comes with TensorFlow, so you don't even have to worry about it if you use Google Colab. Okay. But for those who do want to try to get stuff running on their local computer, I will post links to instructions on how to install TensorFlow. But if you're new, please just use Google Colab. You don't even have to worry about that. It comes with all of this stuff installed. Okay. Okay, so go ahead and add this to your notebook in a cell and import the libraries, okay? So this code here is going to be assuming you're using Google Colab. So you have to say from google.colab import drive, and then we're going to mount to the drive here. Then we specify the path to the data set. Now the data set is a zip file, okay? So we need to unzip it. So that's what these two lines here are doing. It's going to extract that zip file, okay? So we specify the picture size, which is 48, and I'll explain what that's used for in a second. And then here you have the path to the data set, the brain tumor data set, okay? Now this is the data set we just extracted, okay? And just as kind of a side note here, make sure that this data set, this folder here, is in the same folder as this notebook, or else you'll have to specify the full path to this folder, and you can't like do something like this, like just specify the name. Okay, so just make your life easier. Make sure that this folder is in the same directory or same folder as this notebook, okay, as the notebook where your code is, okay? So first we want to visualize MRI scans that have tumors inside of them, okay? So that's what we're going to be doing in this code. So we create a variable tumor presence and we set it to yes, okay? And so we're going to be plotting a figure. Its fig size is going to be, its size is going to be 12 by 12. We're going to be plotting 20 images, so ignore this. This is a typo. We're plotting 20 images. And so we're going to have a for loop here. I is going to go from 1 to 20, because remember, when you make a for loop in Python, the second number here is not inclusive. And we're going to be incrementing by 1. Okay, so here the plot.subplot, this is going to be specifying that we're going to have a grid of images with 4 rows and 5 columns. Okay, so 5 times 4 is 20, right? We have 20 images. Next, we use the load image. We call the load image function. Remember that this is one of the functions that we, oops, wrong way. Sorry about that. So that's one of the functions that we imported here, right? We load image is part of tensorflow.curas.utils. Okay. So we call that, and we pass in the folder path, which is the path to our data set, train, which is the training folder, that's where our training set is located. Tumor presence is yes, because we want images of MRI scans with tumors inside of them. And then this code right here is just getting the ith image in that folder. Okay, so in this case, for the first iteration, I will be 1, so it's getting the image at index 1, okay, in this folder. Then it will add that image to the plot, and then at the end it'll show the plot after it goes through all 20 images, okay? And this is what we see. So here are pictures of MRI scans with tumors inside of them, and you can see that a lot of them are pretty obvious to tell there's something seriously wrong with that brain. Like, you don't have to be a doctor to know that, or to guess that. So, but some of them are a little bit more challenging to tell, like this one, right? Like, is that interference, or are these like tumors, or what is that, you know? So machine learning can really help out here for those, okay? Next, we want to see pictures of MRI scans that don't have tumors in them. So in other words, healthy brains, okay? So we use the same code that we just used, except we changed tumor presence to no, okay? And we print those out, and we see healthy brains here. You can see quite a big difference between the two groups, right? The first group had these big, wider gray blobs, you know, where the tumors were, and this one does not have any of that, right? I mean, they look pretty clean, but a few of them, like this one, you can see some white spots in here. Like, are those tumors, or just interference, or what is that, right? So machine learning can really help in this situation, right? We want to train a model to be able to take in an image and classify whether it has a tumor or not. So you're going to see this pattern, you're going to see this specific pattern here when working with machine learning problems, okay, over and over again. So basically you need 3 data sets. You need a training set, a validation set, and a test set. You're probably like, Ruben, I don't know what those are. No problem, I'm going to explain it. So a training set is what you use, because usually you have like 3 or 4 models that you want to compare against each other to see which one is best, okay, to solve a specific task or problem, okay? And you need a data set to train those models on, right? That's what the training set is for, okay? Next, you have your validation set. The validation set is used to assess the performance of those models you're comparing against each other, right? So you train all of these 3 or 4 models on a training set, and then you get their performance, or in our case, accuracy, on a validation set, okay? So that's what the validation set is for, okay? Lastly, we have the test set. The test set is not touched until the very, very end, okay? Once we have determined our best model, which is the model with the highest accuracy on the validation set, then you take that model and you test its performance on the test set. And that is like the final performance. That's the performance of our best model, okay? The test set is outside data, right? The model was not trained on that data. It's outside data, right? We don't want to test the model on the training set, because that's the data set it was trained on, right? We want to make sure it can generalize, meaning that it can not only do well on the data it was trained on, but it can do well on outside data that it has never seen before, right? We want our model to be able to do that, to generalize well. Okay, so there's some parameters we're going to be passing into here. The first one is the path to the training. In this case, this is a training set, so the path to the images in our training set. So that location is a folder path. Remember, that's our data set, right? That's a variable we specified earlier, /train, because that's where our training images are located. The images for our training set are located, Okay? Then we specify the next parameter is the target size. This is cut off, but it's picture size, comma, picture size, and parenthesis. Okay? So what this is going to do in picture size is 48, if you remember. We set that variable earlier. So this is going to convert every image inside of this folder that we read in. It's going to resize all those images to be 48 by 48. That's what we're telling it to do here, okay? And this next line, the color mode, grayscale, it's going to convert every image to be grayscale if it isn't already, okay? So grayscale images only have one channel, okay? Which, and you might not know what a channel is, that's okay. So an image has 3 dimensions, right? It has width, height, and it has, like, you can think of it as depth. And that depth dimension is the number of channels. Grayscale images only have one channel, okay? Black and white, in other words, black and white images only have one channel, okay? Color images, like for example, RGB images, have three channels because they have 3 channels. One channel for red, one channel for green, and another channel for blue, okay? Grayscale images only store intensity information, so they only have a single channel. Color images store color information, so they need more channels, okay? All right, next we specify our batch size. So when you're working with neural networks, you're gonna pass data. You're not gonna pass a single image at a time through the neural network. That's extremely inefficient. What you're gonna do is you're gonna pass an entire batch of images through, okay? So you need to specify a batch size. So in our case, we're specifying the batch size is 128, okay? Then we specify class mode as categorical. Now, what does that mean, okay? So class mode categorical is going to, let me get rid of this. Class mode categorical, what that's going to do is every image in our data set has a corresponding label. So for example, we have an image, right? It has a tumor in it. Its label is going to be, it's part of the yes class, right? It has a tumor inside of it. Class mode categorical means that our label for that image would look like this, right? And if the image instead was a member of the other class, so it didn't have a tumor in it, it doesn't have a tumor in it, then it would look like this, okay? So that, and these are called one-hot vectors, okay? So one-hot vectors, because you might not know what that means, so is where you have a list with all zeros in it except for a single one, okay? And the one can be anywhere, right? It can be up here. This is another example of a one-hot vector. But in our case, we only have 2 classes, so that's why they're length 2, right? So this would be the label of an image that has a tumor inside of it. This would be the label of an image that does not have a tumor inside of it. And the class mode categorical is what specifies that, okay? Next, we have 2 classes, no and yes, as I've been saying, and shuffle is true. So what does shuffle true do, okay? So as you'll see later, when we are training our model, we're going to train for 10 epochs. So you might not know what an epoch is. That's okay. So an epoch is where you train on the training set from the beginning of the training set to the end one time. That is one epoch. Okay? That's an epoch. And we're doing that 10 times. We're going to do that 10 times, right? When we train for each model. Shuffle true means at the beginning of each epoch, before we train on the training set, we're going to randomly shuffle the training set, randomly shuffle the images in the training set before we do that epoch, okay? And that's because in our case, we don't care about the order of the images. The order of the images are not important. If we were working with like a time series data set, like what we were working with in my other video, the stock prediction one, then the order of the images would be important, right? But in our case, we don't care about order, so we say shuffle is true, and what that's going to do is prevent our model from learning anything about the order of the images, because that's not important, right? And what that's going to also do is help with the generalization of our model. It'll help our model be able to do well on data that it wasn't trained on, okay? Okay. And so that's what these parameters mean, and we're doing that for all three of our data sets, so the validation set and the test set, right? Except obviously we're changing the location of where we're getting our images from, the folder we're getting our images from, okay? Lastly, so this code is going to print out the number of images in each data set. So we have 2,712 images in our train set. We have 288 images in our validation set and 253 images in our test set. Okay, and you can see that we have the highest number of images in our train set, which is good. You want that, right? That means you want your training set to be the largest because that's a data set you're using to train your models, okay? Okay, so hopefully that made sense. All right. And our batch size is 128. Okay, so now we're going to be creating our first neural network. We're making 3 models in this video. This is the first one. This is making a feed-forward neural network, which is the simplest type of neural network that exists, okay? So the first thing we do is we specify the input shape. So like I said, an image is going to be 48 by 48 because remember that's what we're resizing our images to when they're read in, as I just said. And we have one channel because we're working with grayscale images, right? We're converting them to grayscale images if they aren't already. Okay, so we specify that here, and then we flatten it. So we have 48 by 48 by 1 images, but we need a 1D vector. Because feed-forward neural networks expect a 1D vector as input. So you always need to make sure you're passing in a 1D vector into feed-forward neural networks. So that's why we flatten it. Okay, now we have 4 more layers. So the first layer here has 64 neurons in it. The kernel initializer is he-normal. So the kernel initializer is the method that we're using to randomly initialize the weights and biases in this layer of the neural network. Okay, so there's many different kernel initializers. You can look up the documentation and try different ones. This is a hyperparameter you can tweak. Hyperparameters are kind of like the knobs in our machine learning model that you can tweak to try to get better performance, and this is one of them. So you can try different kernel initializers and see what works best for the problem you're trying to solve. Okay, the activation is tanh. Now, one thing I'll say about tanh, you want to be careful when you're working with a deep neural network, meaning a neural network with many layers in it. You want to be careful about using tanh because sometimes it can lead to a problem called the vanishing gradients problem. And I'm not going to get into that. I'm going to make a video later that goes into more detail in neural networks. But basically, it's going to slow down training for you, and that's not going to be good. So in that case, you might want to use ReLU, the ReLU activation function. Just replace this with ReLU, and that might work better in that case, okay? All right, then your next layer is the same except it has 128 neurons. The next one is the same except it has 256 neurons. Lastly, we have our classifier, which has to have 2 neurons in it because we have 2 classes, right? You want to put in an image and you want the output of your neural network to be like this. You want it to have the probability that the image that was passed into the neural network is of class 1, and you want the probability that the image you passed in is of class 2. So in this case, this would mean that the model thinks that the image that you passed into the neural network has a 20% chance of not having a tumor in it and an 80% chance of having a tumor in it, right? And we want our output to be like this. So we need 2 neurons, 1 for each of these, okay? And we use a sigmoid activation function, which will squish the outputs to be between 0 and 1 so that we can roughly interpret them as probabilities. If we didn't have that activation function, these would be just like random numbers, and it wouldn't be between 0 and 1, so it wouldn't be as readable, okay? Lastly, we're going to use an ADAM optimizer with a 0.001 learning rate. So ADAM is the algorithm that we're using to train our neural network, and we're using a 0.0001 learning rate. Now, this is like the most important hyperparameter in a machine learning problem, okay? You definitely want to take the time to tweak your learning rate to figure out what works best for the problem you're trying to solve. Next, we're compiling our model. We're using the categorical cross-entropy loss function. This is the loss function that your model is trying to minimize during training, and you can experiment with different ones. We're using the ADAM optimizer, like I mentioned. And lastly, we get the model summary, which will print out this. This is our model summary, and it shows you the output shape of each layer and the number of parameters, aka the number of weights and biases in each layer, and the total parameters, number of trainable parameters, and the number of non-trainable parameters in your model. Okay, now we're going to train our model. So we're going to pass in the training set because this is the data set we want to train our model on. Then we specify the steps per epoch. You can just copy and paste this code into your project. This is cut off as train set.batch size. That's what this is supposed to say. But for an example, let's say the training set is size 25. Okay, we have 25 images in our train set. And let's just say, as an example, our batch size is 5, okay? So it's going to be 25 divided by 5, which is going to be 5. That's what will evaluate to. And that means we will take 5 steps in order to get through our training set for one epoch, right? And each step is a new batch that we're training on. And each batch is going to have 5 images in it, right? So we're taking 5 steps that'll get us through the whole data set. And each step will be a batch of 5 images, okay? So that's what that is for. Next, like I said earlier, we're training for 10 epochs, and I already explained what an epoch is. And we pass in the data set we want to use to evaluate our model, which is the validation set, okay? And history 1 will store the accuracy of our model on the training set and validation set for each epoch. This is history 1 will contain what this function returned, what this method returned, okay? Okay, so this is what the code I just showed you will output, and you can see so it contains the accuracy of our model on the validation set, the loss of our model on the validation set, the accuracy of our model on the training set, and the loss of our model on the training set for each epoch, after each epoch, okay? And what you want to see is that your accuracy gradually increases and your loss gradually decreases. If you see that, then you're training well. If not, then you need to go back and tweak hyperparameters to try to get it to train better, okay? So you can see that we started at about 61% accuracy on our validation set, and we end with about 73% accuracy on our validation set after 10 epochs of training. So that's a pretty good jump there, right? And we can see that the accuracy on our training set starts at 55% and ends at about 76%, okay? All right. Next, we're going to plot this data that I just talked through, and this means we're going to be plotting the accuracy of our model in the training set for each epoch. And this means we're plotting the accuracy of our model in the validation set for each epoch. Then we specify the title of our plot, model accuracy. The Y label is accuracy. The X label is epoch. We have a legend with train and validation in the upper left, and we show the plot. That will output this plot, okay? And what you want to see is an upwards trend, right? As you train for more epochs, your accuracy should increase, right? And one thing you need to be careful about is if at some point these two lines start to diverge and you end up with like a much higher training, a much higher accuracy on your training set than your validation set, that indicates that your model has overfit. Okay, and what that means is that so basically the model is learning patterns in the training data when it trains on it, and it learns these patterns and uses those patterns to be able to classify images it sees as having a tumor or not having a tumor. If those patterns that it learns don't help it classify images in data that it hasn't seen before, that's not good, right? Because if it overfit, that means the patterns it learned are only good for classifying images in the training set. That's not good, right? Because when we use it to classify images in data it's never seen before, it's not going to do well because those patterns that it learned aren't helpful to allow it to generalize. So if you do overfit, which you'll be able to tell by what I just described, then you need to tweak your model, use some regularization methods to try to get your model to not overfit. Okay? So hopefully that makes sense. Okay. In our case, they're close, so we're not overfitting here. Okay? Okay, so we got like 73% accuracy on our validation set. That's pretty freaking terrible for a model that's going to be used to detect brain tumors, right? A lot of people are going to die. So that's not good. We need to get better accuracy. We need to do better. So why did that not do so well though? Like why did the model we just trained, why did that not do well? One possible reason can be local spatiality. Since these tumors are present at varying locations within these MRI scans, feed-forward neural networks like the one we just trained wouldn't be able to adjust their weights to detect their presence at a particular location within the image. We can try convolutional neural network models to resolve the issue due to local spatiality. Okay? So I'm going to be explaining this first convolutional neural network model. I have one more after this and making it so you can understand it, okay? So first, we start by creating a sequential model using Keras. The sequential model is a linear stack of layers that allows us to create a neural network very straightforwardly. Next, we add our first convolutional block, this is our first convolutional block, which consists of a conv2D layer followed by a max pulling 2D layer. Okay, so I'm going to get a little technical, but after I say the technical details, I'm going to explain it so it's clear to understand. So don't worry, okay? So conv2D stands for a two-dimensional convolutional layer. This layer creates a convolution kernel that is convolved with the layer's input to produce a tensor of outputs, okay? So what does that mean? So this layer has kernels, things called kernels. These kernels are going to do this special operation called the convolution operation on the input of this layer to calculate the values that are the output of this layer, okay? So more specifically, in our case, it will learn 16 filters, each being a 3x3 window that will be applied on input images. Each filter represents a feature detector, like edges or textures, that enhances relevant parts of the input. The ReLU is an activation function that introduces nonlinearity into the model, enabling it to learn more complex patterns. The input shape specifies the dimensions of the input images, which in our case are, like I said before, 48 by 48 grayscale images. So we have one channel because it's grayscale. Max pulling 2D is a layer that effectively downsamples the input by taking the maximum value over the window defined by pool size, in our case the pool size is 2 by 2, for each dimension along the feature axis. Here it reduces the spatial dimensions height and width by half. Okay, so what does this do? So this has max pools in that layer, and what it's doing is it's cutting the size of the input neurons. And when we're talking about convolutional neural networks, we call neurons feature maps, okay? So it's reducing, because in our case, we specifically have a pool size of 2 by 2. So what it's doing is it's cutting the size of each of our neurons or each of our feature maps in half, reducing its size by half, okay? And what that's gonna do is reduce the number of parameters, which is gonna make training easier. And it's gonna help our model to generalize, right? To do well on data that it hasn't seen before, okay? Or hasn't been trained on, all right? So we repeat this structure to create a second convolutional block. This block is similar to the first one, but it learns 32 filters. As we go deeper into the neural network, layers tend to learn more complex, high-level features, okay? The flattened layer then transforms the 2D matrix, so height, width, and channels, into a one-dimensional vector, okay? So like I said before, feed-forward neural networks expect a one-dimensional vector. And you might be like, Ruben, what the hell? Like, I thought you just said we're doing convolutional neural networks. Why are you talking about feed-forward neural networks? I'm confused. Don't worry, okay? So convolutional neural networks, at the end of them, usually contain a feed-forward neural network. And that's what does the final classification, okay? So we have to make the data a 1D vector so that we can pass it into the feed-forward neural network. And that's what this flattening layer is doing. Okay, so afterwards, so that now we're getting into the feed-forward neural network. So we add a dense layer here, also known as a fully connected layer. Okay, and it's called a fully connected layer because every neuron in this layer is connected to every neuron in the previous layer. Got it? This layer learns from the features extracted by the convolutional layers to make the final prediction. It has 128 neurons and uses the ReLU activation function. The final layer is also a dense layer with 2 neurons, corresponding to our 2 classes, right? Like I said earlier, we need 2 neurons because we have 2 classes. The sigmoid activation function squishes values between 0 and 1, like I said earlier, making it suitable for binary classification, right? So after defining the architecture of the neural network, we need to compile it now, okay? So here we define the optimizer and the loss function. The optimizer updates the model's weights based on the data it sees and the loss it has incurred. Atom is a popular choice due to its computational efficiency and good performance on a wide range of problems. The learning rate here dictates the speed at which the model learns. The categorical cross-entropy loss function can be used when we have a binary classification problem. It measures the performance of a classification model whose output is a probability value between 0 and 1. Finally, we print a summary of our model to check if everything is as we expect. Great. Okay, so now let's see the model summary that we printed out here. So let me just say one thing that's very important to kind of like the goal, one of the goals in a neural network, in a convolutional neural network, is to gradually reduce the size of your neurons or your feature maps until it's a 1D vector and can be passed into your neural network, right? So we start with 46 by 46 size feature maps, right? And we're gradually reducing it. So we go from 46 to 46, 23 by 23, then we go to 21, 21, and then 10, 10, right? Until we get it to a 1D vector, and then we can pass it to our feed-forward neural network, which will make the final classification, okay? Then we train it using the same code as before. And this time we start at 72% accuracy on our validation data set. And we get up to 81% accuracy. So a pretty good jump there. And for in terms of our training set accuracy, we start at 59% accuracy and we end up at 87% accuracy. That's a good jump, right? So, and we also see that our accuracy gradually increases and our loss gradually decreases, which is good. We're training well, right? So that's good, right? We have increased to 80. Now we're getting 81% accuracy, right? So that's much better. So now we're going to use the same code as before to print out the same plot as we showed before, right? Except for this model now. So now we see that as we train for more epochs, the accuracy of our model on the training set and the validation set increases, which is good, right? We want to see that. Now you might be a little worried because, as you can see, like if you remember what I said about overfitting, these are kind of splitting up, right? Diverging. But in this case, I don't think they're far enough apart to really say it's overfitting. You know, we ended up with 81% accuracy on the validation set and 87% accuracy on the training set. So I don't think that's a big enough difference. I wouldn't say it's overfitting. So we're good here. Okay, so we got 81% accuracy now, okay? So that's better, but still a lot of people are going to die. That's not good enough, right? We need to do better. So let's make a better model, right? So that we can save more lives, right? So that's what we're going to be doing now. I'm going to be explaining the architecture of this model. Okay, so we start with creating a sequential model. This type of model allows us to stack layers on top of each other in a sequence, which is the most common way of building neural networks. We then add the first convolutional block. The Conv2D layer applies 64 filters, and I explained how these work earlier. Each of them are size 3x3 to the input image. We're padding the input data using same padding. So this is new. I haven't explained this yet, so let me jump ahead for a second so I can explain this. So if a layer has padding equals same, what that means is the feature maps that the size of the feature map that comes into the layer, which for this layer, the input is 48 by 48, right? Because we have 48 by 48 images, is going to be the same as the size of the feature maps that come out of this layer, because we use padding equals same. And this layer here also uses padding equals same. You can see that what comes in is 24 by 24 feature maps. And what comes out is also 24 by 24 feature maps, right? So that's what padding equals same does. So more specifically, it means that we add zeros around the input image to keep the output size the same as the input, okay? The input shape indicates that our input images are 48 by 48 pixels with one color channel, because it's, grayscale. The activation ReLU adds a non-linear transformation called ReLU, rectify linear unit. It's an activation function, okay? Which turns all negative pixel values to 0. That's what ReLU does, okay? Finally, the max pooling 2D layer reduces the spatial dimensions, height, and width of our intermediate feature map by taking the maximum value of our 2x2 pooling window. And I already explained how max pooling works. The same process is repeated for the second, third, and fourth CNN blocks, but with more filters, as you can see here, and larger filter sizes, right? 3x3, and now it's 5x5 in the convolutional layers. After we've passed the data through all the convolutional blocks, we flatten it. So let me go to the next page. So now we flatten it. This is a continuation of the previous code, all right? So now we flatten it. And remember, we have to flatten it into a 1D vector so that we can pass it to the feed-forward neural network to make the final classification, okay? So this takes our 2D feature maps and turns them into a 1D vector. Next, we have the fully connected or dense layers. This is the feed-forward neural network now. That's what we're getting into now. These layers perform the high-level reasoning in our network. Each dense layer performs a linear operation on the layer's input, followed by another ReLU activation. The last layer of our network is the classifier, which is here. Here we're using two neurons. This is the same as I described earlier. We're using two neurons because we have two classes to predict. The softmax activation ensures the output values are probabilities and that they sum up to 1. Each neuron will output the probability of a given class, like I showed earlier. Finally, we compile the model. This means that we're ready to start the training process. The model will use the Atom optimizer with a learning rate of 0.0001 to change the weights and biases in the network. And it will use the categorical cross entropy loss function to measure how well the model is doing during training. Awesome. Okay, so now let's print out the summary of this model. You can see we're gradually reducing the size of our feature maps. We start out with 48, 48, then reduce it to 24 by 24, then 12 by 12, all the way until we flatten it into a 1D vector, and then we can pass it to our feed-forward neural network, which will make the final classification for us. Okay? Fantastic. Okay, we train it using the same code as before, and we get 100% accuracy in our validation set. That's awesome. So I rounded up, right? It's 99.7% accuracy. But that's phenomenal. And we get about the same accuracy on the training set. We can see that our accuracy gradually increases, our loss gradually decreases. So that's good. We're training well. And we start at 78 after epoch 1, and then by the time we finish epoch 10, we're all the way up to 100% accuracy. Awesome. We use the same code as before to plot it, and we get this beautiful chart showing us that the accuracy of our model in the training set and the accuracy of our model in the validation set gradually increase as you train for more epochs. Awesome, and we're not overfitting, right? Great. So now we have our best model, right? Obviously, the one we just made is our best model. I mean, we got 100% accuracy on our validation set. So that's the one we choose as our best model. So now that we have our best model, remember what I said. Now we grab our test set. We kept it aside all the way until the end. So we have our best model. We grab our test set. And now we're going to test the performance of our best model on that train set to get the final, performance of our best model, okay? On data it hasn't seen before, right? Data that it wasn't trained on. Okay, so that's what this code is going to get the next batch from the test set. So remember, our batch size was 128. So we're going to get 128 images and their corresponding labels. And we are going to call this method evaluate on the model and pass in the images and labels from our batch. And then verbose 1 is just going to print out this stuff that you see here. And we get a final accuracy of our model on the test set, which is 99.2%. That's phenomenal. That's great. Okay, so now we're going to get the performance of our best model in more detail. Okay, see how good it performs in more detail. So we're going to do this by first we say model 2, which is our best model, dot predict, and we pass in the images from the batch we just got, okay? And this is going to predict whether, for each image, it's going to predict whether it has a tumor in it or it doesn't have a tumor in it, okay? But we have one problem, so remember what the output of the neural network is going to be, right? It's going to be like this. It's going to have it's going to be like, it's going to say, okay, this image has a 20% chance of not having a tumor in it and an 80% chance of having a tumor in it, right? We don't want it to be in that format. We want it to be just zeros and ones. So like an image we have an image and its label is 0, and we have another image its label is 1, right? We don't want labels to look like this. So how do we do that, right? So what we need to do is convert a label like this to be just 1, right? Because which index is higher, right? This is index 0, this is index 1. Which number is higher? This number is higher. And what index is that? 1. So we want the label to just be 1. We don't want all of this. So how do we do that? Well, we use np.argmax. This is a NumPy function called argmax. And we do that over the first axis. That's going to convert all of the labels that look like this to just being a number for each image, okay? And then we do something very similar for the test labels. Now the test labels are a bit different. Instead of being probabilities, they're just going to be a one-hot vector like that. So if the label for an image is yes, meaning that it does have a tumor inside of it, then the label would be this, right? Because this index indicates no, that it doesn't have a tumor in it. This index indicates yes, that it does have a tumor in it. Okay, so if let's say we have a different image and it doesn't have a tumor in it, right? So then its label would look like this, okay? So that's how the test labels look like. So we need to convert those to numbers as well. So this should become 1 because this index is higher, and this should become 0 because the 0th index is higher, okay? And we can use the same function, right? Because we just want to return the index that has the highest value. So we use the same function, np.argmax, over the test labels, axis 1. And now we have our predictions as just zeros and ones. So each image either has a 0 or it has a 1, indicating what class it belongs to. And these are our predictions, right? So if we predicted that an image does not have a tumor in it, it would be 0. And if we predicted that an image does have a tumor in it, it would be 1. And why true? These are our ground truths. So this will contain whether or not each image actually has a tumor in it or not, right? It's our ground truths. Okay, so if an image actually does have a tumor in it, it would be 1, and if it doesn't, it would be 0. Okay, and now that we have our predictions and like the ground truths, we can see how good did our model perform, right? So first, we're gonna print out the classification report. We pass in Y true and pred. We're not really gonna focus on that for this, but we're printing it out. And then we print out the confusion matrix, which is also known as the classification matrix, by passing in Y true and pred. Okay, and this code below it just makes it look pretty, basically. Okay. Now what's up above is going to be the classification report, and this is the confusion matrix. Okay. The thing below this is the confusion matrix. So the worst possible scenario for this problem is where we predict no, but the actual is yes, meaning we predicted that an MRI scan does not have a tumor in it when it actually did. Because then we're in a clinical trial, we're basically telling someone, hey, you don't have a tumor, but they actually do, which could kill them because they're not going to get the treatment they need, so they could die, right? Because with tumors, you want to catch it as early as possible. So we want this number to be as low as possible, okay? This absolutely needs to be as low as possible. In our case, it's 0, so that's the best you can possibly do. So we did phenomenal, right? This is not as serious, but we still want it to be as low as possible. What this means is that we told someone, hey, you do have a tumor, but they actually don't, okay? So yeah, we for an MRI image, this case is that for an MRI image, we predicted that it does have a tumor in it, but it actually doesn't. That's not as serious because we could just, maybe we give them some medicine, but in the next stage of the clinical trial, we'll catch our mistake, and they'll be taken off the medication. They're very likely to be perfectly fine. So this is less serious than this, but we still want this to be as low as possible. In this case, it's just 1, so it's really good. We did very good. And then these 2 squares along this diagonal here are just correct predictions. So we predicted that an image doesn't have a tumor in it, and it actually doesn't. That's this case. In this case is that we predicted that an image does have a tumor in it, and it actually does, right? So to read this, it says 80, so that would mean that there were 80 instances where we predicted that an MRI image has a tumor in it, and it actually did. That's how you can read this, okay? Okay, so obviously this is a phenomenal model, right? This is so good it could actually be used in a hospital, okay? All right, and that's all. Thank you so much for watching. I really appreciate it. I love making these videos for you, and it takes me a lot of effort to make these videos, so it would be awesome if you could show your appreciation and like and subscribe. That would be awesome. Thank you so much, and I look forward to seeing you in my next video. Bye.